Today we're going to be talking about frontiers of cell therapy, and it's my pleasure to introduce our three speakers, but I thought I'd take just a minute to sort of set the context or stage for what we'll be talking about. And I'd do this maybe in an odd way by making you think about your cell phones and your computers, which is the transistor was discovered in the 1940s and 50s, and it took a long time before cell phones or computers were developed after that. In the history of medicine, if you think about how many diseases have been cured in 1 or 2 years, that number is 0. It takes a long time to begin with a new platform technology and develop into an effective therapy. And today you'll hear from 3 people who are really committed and experts in that general area. Stem cells were discovered in mice decades ago, in humans in the 1990s, and some people are saying, well, where are the cures? How come we didn't already have a cure? And the fact is, people like you'll hear from today are being very strategic and careful about going through all of the steps to make sure that a cure is safe and effective. And we're going to hear about particular diseases, some ideas about where the field may go, and also address issues of how do you actually go from an academic discovery, which we're hearing quite a lot about at this meeting, to a product, what's involved in manufacturing and delivery. So it's my pleasure, as I've already indicated, to introduce you to 3 experts. I'll introduce you to them now, and then they'll speak for a few minutes, 5 to 7 minutes, with or without slides, in order. First, I'm pleased to say we have Dr. Masato Takahashi here from the Riken Center for Developmental Biology. She'll be speaking about eye disease. I'm also pleased to welcome Molly Choiquette. She's the University Professor of Chemical Engineering and Applied Chemistry from the University of Toronto, and she'll be speaking about applications in a number of disease areas which involve both cell manufacturing, scale-up, and delivery. And finally, my friend and colleague from Harvard, George Church, Professor of Genetics and Health Sciences and Technology, and I'm not going to tell you what he's talking about because he always surprises me with new ideas. So let's begin with Professor Takahashi, please.
Thank you. My name is Masato Takahashi from Riken, and I've been working about the retinal cell therapy using iPS cells. And I started my research about the stem cell in 1995 when I was at Salk Institute in the Dr. Gage's lab. And finally, we reached to the clinical application. And since this year is the 10th anniversary of the human iPS cells, and I'm glad that I can report this successful application of the iPS cells. In 2014, September, we did the first surgery using iPS cell-derived cells. At that time, we did the autologous transplantation of human iPS-derived retinal pigment epithelial cell sheets. We took the skin fibroblasts from patients of age-related macular degeneration, and we made more than 30 lines of iPS cells. And we chose the beautiful iPS cell lines by mainly morphology at the time, and make them differentiate into retinal pigment epithelial cells. Retinal pigment epithelial cell is derived from neuro-2, but it's a special epithelial cells that maintains the photoreceptor cells. And we purified the RB cells, and for the first patient, we've got 100% pure retinal pigment epithelial cells. And we let them make the basement membrane by themselves and transplant the cell sheet beneath the patient's retina. And after 2.5 years, still the cell sheet survived and functioned very well. There was no immune rejection. Of course, it's because it's an autologous transplantation. Although it was really successful and we confirmed the safety of iPS-derived cells, but the autologous transplantation, the cost is very high and time-consuming. We could treat only 1 or 2 patients in a year. So from the beginning, we planned to move to the allogeneic transplantation. Meanwhile, Dr. Yamanaka in Kyoto University he made a plan of the iPS cell stock. And they made the special iPS cell line with HLA-6 loci homozygous person. And with this most frequent pattern combination of the HLA-6 loci, only one cell line covers 17% of Japanese population. So we can treat many patients with this only one cell line. And we did many experiments to confirm there will be no or very small immune rejection using this HLA-matched transplantation. And last March 2017, in March, we did the first transplantation of using allogenic iPS cells with little immunosuppression. We did only one injection of the steroid at the end of the surgery. We are planning to do 5 patients in this clinical study and see the immune reaction to those cells. We did the first application using iPS cells. It was because we chose the right cell, very safe and very good characteristic cells. And also in Japan, we have a special system for the regenerative medicine. We have these 2 laws, one for clinical research, it's called Act on the Safety of Regenerative Medicine, and one is for clinical trial, mainly for the company. It's a Pharmaceutical Affairs Act. And we use the track of clinical research. iPS cell treatment is a developing treatment, so we should modify every protocol in several years, I think. So for this reason, the law for clinical research is very suitable for the cell therapy, so that we could do the first clinical application, I think. So we will report the results maybe within 2 years or so about the allogenic transplantation. Thank you very much.
Okay, good afternoon, everyone. It's a great pleasure to be here. Being Canadian, I like to follow rules, and we were actually given instructions in terms of what the panel will present. And given that my association is in engineering and approaching the field of regenerative medicine from that perspective, I thought I'd highlight some of the challenges we face in regenerative medicine around cell expansion, cell survival, and cell integration, and then highlight some of those issues related to engineering and drug delivery. So I'm going to highlight a couple of companies and then go into some more of the basic research. So XcelThera is a new company, and it's based on 2 technologies. One is the discovery of a small molecule, UM171, which results in about 100 times expansion of hematopoietic stem cells, maintaining CD34 positive cells. And then another, which is the use of a bioreactor to get a 10 times expansion of these cells and a 50 times expansion of the CD34 cells. So these are 2 innovations that come together. And this company is working obviously focused on cell expansion of the HSCs from umbilical cord blood. And the first focus is on acute myeloid leukemia, but they'll also focus on other blood diseases. Another big challenge we have in the field is around cell survival and cell integration when we're transplanting cells. And so I wanted to highlight Blue Rock Therapeutics. As you know, Blue Rock has one of its centers is in Toronto. And that is really a reflection of the work of Gordon Keller and Michael Laflamme, where they're working on ventricular cardiac muscle cells for use after myocardial infarction. And then it also brings from New York, Lauren Studer's work on pluripotent stem cell derived neurons for the treatment of Parkinson's disease. Around cell transplantation, some of the work now going into my own lab is really, can we enhance cell survival and cell integration? Because one of the things that we've observed over the past several decades is that most of the cells that are transplanted don't survive well. And so we've invented a hydrogel, just an injectable material that we've shown promotes greater cell survival and integration. And so what you can see in this slide on the left is the cells injected in the hydrogel, and this is in the subretinal space, and it's a collaboration with Derek Vanderkooy's lab. Those cells are spread out more evenly than the ones that are all clustered and aggregated together in the center panel when those are delivered in traditional media, just like saline. And one of the things that we realize is while we've got more cell survival and integration, still it's a very low percentage. And so we're thinking now of how can we enhance the environment into which we transplant cells to get greater cell survival and cell integration. And so the figure on the left is really work ongoing in the lab. We're also really interested in cell stimulation. So we know that if we can stimulate the endogenous stem cells, then maybe we don't have to transplant them. And so our lab has been focused on the central nervous system and designing strategies that will achieve local delivery to the CNS. So as you know, the blood-brain barrier, the blood vessels in the brain and the spinal cord limit the diffusion of many protein therapeutics into the central nervous system, and thereby requiring us to come up with more innovative strategies to stimulate the endogenous stem cells in the brain or the spinal cord. And so through a number of different collaborations, we invented, again, an injectable biomaterial that we can place on top of the tissue, allowing the protein therapeutics to diffuse into either the brain tissue or the spinal cord tissue. And through this process have invented a series of different biomaterials and delivery strategies. And lastly, one of the things we've heard a lot at this meeting is the use of 3D models of disease, and can we grow cells in a 3D environment to better understand disease progression and then ultimately use that to come up with better therapeutics. And again, approaching this from the engineering perspective, what we've done is come up with a defined matrix, defined hydrogel that we hope will replace Matrigel. Matrigel's been a great material for everybody to study, but it's inherently limited because it is not defined. And so a lot of what we're learning by the use of Matrigel and 3D platforms, I think, can be misleading. And so these are just with breast cancer cells, and so we've looked at that with a number of different systems. And so lastly, I just highlight the ecosystem in Toronto, where we are starting to take advantage of what's going on in the hospitals and the academic institutions. We've had a lot of investment in the field of regenerative medicine through Medicine by Design, the Ontario Institute of Regenerative Medicine, the Centre for the Commercialization of Regenerative Medicine. So a lot of these acronyms, but that is then leading to new companies, and some of those are highlighted here. Thank you.
Yeah, I just want to focus on some recent progress that we've made that I'll be talking about tomorrow in more detail, but in particular ways that we can do genetic coupled with epigenetic modifications of cells intended for either therapy or testing therapies. But let's focus mostly on the therapies. When we make genetic alterations, as you heard a little bit, maybe if you were present at the briefing early this morning, we have problems with on-target and off-target errors that are made in the genetic manipulation. We have found that if we do clonal analysis, if we have the luxury of developing a large amount of tissue, as we just heard, expansion being the challenge here. If we can go from single cells, then we can make sure that we have perfect on-target and off-target. To put this in perspective, the off-target error rate can be as low as 1 in 10 trillion. That's on a base pair level, but if that involves treating many millions of cells, you still have a potential off-target issue. But from a clonal analysis, it's 10 to the minus 13 times the precision of the cloning and sequencing, cloning of the cells and sequencing, which is another 6 orders of magnitude. In terms of delivery, survival and delivery, which are some of the challenges here, we have progress in making organoids and organs. The organoids are a modest way of saying that we're not quite to the point of having perfect organs that are transplantable, but this is one way of delivering cells that are either clonally derived or possibly non-clonally derived. So we've had great progress in this. We have a paper that's under review right now that I will talk about tomorrow, where we've finally have all of the human transcription factors, at least one transcription factor per, one expression clone per transcription factor gene for humans. And we've tested these singly in various combinations. We have a pipeline where we can get essentially every cell type, at least in broad strokes, that we've attempted to derive, about a dozen so far. And we're moving towards, for both the genetic and epigenetic reprogramming and this transcription factor library, we want to ask how precisely these are dealing with the pathological state, how precisely they're representing a natural state, and so forth. And for this, we've developed a new technology, which was launched as a company called ReadCore. The technology is fluorescent in situ sequencing, and this allows us to assess DNA, RNA, and protein levels at the single molecule level, at the subcellular resolution, so we can assess whether the therapies, the organoids, what have you, are how they compare to a corresponding natural human tissue. So I'll turn it back over to Doug.
Before we take some questions, maybe I'll try to synthesize what we've heard and provide a bit of an overview. I'll go sort of in reverse order. So George talked about the grand vision of, can biologists actually gain control over the code that tells cells what their fate should be? And this involves both concerns about safety, but as he emphasized, changing the genetic modification and the epigenetic modifications of cells with the ultimate goal of controlling cell fate, telling cells what to do. That is broadly applicable to practically any disease one can think about. Molly very nicely talked about a couple of important problems. So let me give an overview of that in the following way. When one thinks about cell therapy, we either are thinking about stimulating endogenous stem cells to sort of get off the couch and help repair the body, or to correct their behavior in some way. Or, as Molly was describing, we're thinking about growing cells outside of the body and then putting them back in. For endogenous cell therapies, one doesn't think so much about cell number, but if you're thinking about diseases where you're going to repopulate the body with cells, you have to expand them under safe and effective conditions, and that's really where chemical and biological engineering come to play. Delivery then is another issue. Molly also mentioned the possibility of direct injection, putting cells in like little jello matrices, trying to figure out how do you put the cells where you want them. And then this brings me to Dr. Takahashi's talk, which is just to remind us that depending on the disease, it is or is not important where you put the cells. So, for example, Dr. Takahashi talked about putting retinal pigmented epithelial cells in the eye. It's sort of laughably silly to say, well, why not put them somewhere else? Well, you can't put them somewhere else because you want the eye to work. Similarly, Molly talked about Parkinson's disease, where you have to put the cells where they would normally be in the midbrain in order to have a function. I work on a disease where you put the cell doesn't matter. So this is diabetes, and I'll remind you that you can inject insulin pretty much any place in the body. So once one is able to do the things they talked about with a different kind of cell, a pancreatic beta cell, you don't have to put it back into the pancreas, or you don't have to put it into a particular structure. That cell will function anywhere in the stomach, in the buttocks, in the legs, subcutaneously. And so that is a sort of different challenge. But again, Molly reminded us that in any of those cases, you have to expand the cells and do it under safe conditions. Make sure that you're not getting, as George would have indicated, genetic modifications that would produce tumors, or the cells would sort of go off path and do something that you don't want them to do. I often think about this like if you're trying to get, say, let's say from Boston to Toronto. You want to get there on the right path, and you don't want to take a wrong turn and make the wrong kind of cell and end up in Chicago. So this is really the challenge for the field, which is, depends on the disease, are you going to use endogenous or exogenous cells? If it's the latter, you have to expand them. You have to be very careful about the genetic and epigenetic control, and there George Church's lab has been leading the world in how to think about really gaining mastery and control over the genes that then allow us to control cell fate. Now, I don't know that my sort of off-the-cuff summary was very helpful, but let's see if it stimulated any questions from the outside. And the questions can be directed at any of our speakers. Yes. Antonio, may I ask you to, does Antonio need to come to the microphone, please? Yes, thank you.
My name is Antonio Regalado, Technology Review. Dr. Melton, you mentioned, you made a comparison to the transistor leading to the smartphone, but of course transistors had utility, practical utility almost on day one. They didn't have to wait for the cell phone to come. So I take issue a little bit with the analogy, but maybe you can make some more comments because Molly's slide of all the companies, it seems like there's maybe kind of, is there a renewed activity by companies in this space? I mean, give us a flavor for the appetite that people are putting money on the line have sort of currently for IPS technology, say compared to yesterday or a year ago.
Yeah, well, you and I could argue about the history of the transistor, but I'd suggest to you it took about a decade before there were transistor radios. And stem cells have been useful from day one for research purposes. But the import of your question, and thank you for it, is, well, as a patient, I don't care about what they're doing in research labs. I want to know, when is there a product that makes a difference? And the point I was trying to make, perhaps clumsily, was that one shouldn't expect a product within a year. One should better think of it more in terms of a decade. And Dr. Takahashi has given us really the first example of that. I think she nicely said, am I right in thinking it was about a decade? Yes. 2 decades from the initial discovery. We all hope in this field that that time will get shorter, both by the investment of more capital, but also by know-how. If Dr. Takahashi were going to start today, as she said, it wouldn't take her 2 decades to do the next patient with a different type of cell, because there's been so much learned in the process. A more expansive answer to your question about companies is, yes, I think you will see increased investment both in small biotech companies that are starting, and you heard some in Toronto. George mentioned one. I like to think Boston is one of the hotbeds of that activity. I don't know how many companies are here, but it's a significant number. And greater interest from large pre-existing pharmaceutical companies is on the rise. Most of the large companies that I know about now have some kind of stem cell program, an IPS program, maybe for drug discovery, where you're not thinking, and again, I'll go back to Molly's talk, you're not thinking about making cells that you would put in the person. You're thinking about making cells from a person that has a disease and a person that doesn't as the control, and then doing a drug screen and asking what's wrong with the defective cell. So I think you probably know this, Antonio, but I'll amplify with a specific example. At the Harvard Stem Cell Institute, Kevin Egan and others have made iPS cells from people that suffer from amyotrophic lateral sclerosis, Lou Gehrig's disease, and from normal patients, made motor neurons, which is the cell defective in the disease, then screened for drugs which slow the defect in the motor neurons, and that drug is now in a clinical trial. I don't know how long that took. I don't think it was quite a decade, but it was close to a decade. And so I'll finish by saying that I accept your sort of challenge is what's taking so long, and is this going to come to bear? But remember, we're not talking about making a new phone app like Snapchat. We're talking about something that goes into people and will affect their life forever. So let's do it properly and carefully. Molly, did you want to add something? Yes, please.
Yeah, I'll just add that I think that the investment from Big Pharma has been somewhat cyclical, and I think we are in a good place right now in terms of that investment. I mean, we saw with Blue Rock Therapeutics, I think it was the second largest Series A, $225 million, a joint venture from Bayer and from Versant Ventures. So that's certainly a big investment in this field. And I think that we are seeing pharma come back in. I don't know if it's decades, but they do. I do think it has been somewhat cyclical, and I think we are in a great opportune time now where we are seeing more investment.
George, would you like to comment on capital investment in the field? Because you've had non-trivial experience with startups and large pharma.
Yeah, well, now is I think, a particularly good time. There's a lot for people trying to do translational medicine, trying to get investment in good ideas. They have to be good ideas. Some, including things that are not mature and things that are already, that were within a year or two of preclinical trials. Our most recent ones were Egenesis, which was not quite a cell therapy, it's more of an organ therapy by developing organs in pigs. The ReadCore I mentioned, which is for analyzing cells for therapy. And of course, Editas, which is editing cells for therapy, which goes much further back to Sangamo for editing T cells and so forth for therapies. So it's in a way it's a mature field, but I think within the last 2 years I've seen a huge increase in enthusiasm for both the cell therapies and genetic modification.
Yeah, it reminds me to say something that's perhaps obvious, that if you ran a drug screening company and for decades you've been screening on, say, rat fibroblasts to find things that affect diabetes or Parkinson's, an undergraduate would say, well, why not make a human cell that is affected in the disease and do your screening on that? And that's really the shift we're seeing. But it doesn't happen in a year. It takes a long time to shift both personnel who know about how to do this and to shift divisions within large companies to work in the area. And we're seeing that shift occur now. Other questions, please? Yes.
My name's Drew Joseph. I'm at STAT. I have two questions, actually. One for Dr. Takahashi. Is it sort of with mutations when using iPS cells, is it the same concerns using autologous versus allogenic, or are there different concerns? And then I have a second question.
About the genetic mutation, the original mutation and the acquired mutation have different meanings. And
The acquired mutation.
Okay, and the acquired, of course, it matters, but it depends on what kind of end product you have. For example, the retinal pigment epithelial cells, even though they have the P53 gene mutation, that's a strong oncogene mutation, still they don't make any tumors at all. But hepatic cells is different, so you should think about the what kind of end product you have. And of course, but it's better to have no mutations. And in that sense, the allogeneic transplantation, the cell line can be thoroughly evaluated about the gene mutation. So of course, the allogeneic will have more safe without any gene mutation, I think.
Before you ask your second question, sorry to interrupt, I want George to see if he can answer a question that I've wondered about. The mutation issue, people worry about, because mutation usually sounds like bad, right?
Yeah,
mutation is something
right.
you don't want. But if you were to take, say, cells in my body and do whole genome sequencing on them all, how many cells out of 1,000 are, quote, unmutated? Is that known?
It is partially known. I look forward to it being very well known, but There's on the order of a few mutation, or 0 to a few mutations per cell division. And so basically no two cells in your body are identical as far as we know. Obviously we haven't proven that one by counting. But there's about 30 plus changes per generation in germline. And if you look at the number of CRISPR off-target mutations, it typically is less than the number of mutations you get due to somatic variation. During the number of cell divisions it takes to do the analysis, the mutation and analysis of the CRISPR, you've got more mutations due to replication than you do to off-target.
Yeah,
if it's been a well-designed and well-vetted guide RNA, that is.
It doesn't change the import of your question, because you really meant mutations that cause harm. I'm just sort of reminding us that mutations are a natural function of cells dividing and being alive. And as Dr. Takahashi said, they're very attentive to ones that would cause harm.
As a rule of thumb, that you can estimate the fraction that will cause harm if they're randomly distributed, because about 1 to 2% of the genes are tumor suppressors, which is, if you're going to have a null that's even heterozygous, that puts you at risk, but it doesn't necessarily cause a tumor, so that's 1%. And about 1% of the tumor suppressor gene is exons, which are susceptible to mutation or other parts. So it's about 1% of your random mutations predispose you to cancer without actually causing it, and then another even smaller percentage will cause it swiftly.
So sorry, I didn't mean to say your question was, I was just putting your question in
No, I
context.
appreciate
It's a very good question.
The second question is, with 21st century cures, there's already been some changes to the regulatory landscape for cell therapies, and I'm curious to know, as sort of like careful scientists, but also people who want to get things into the clinic, how do you feel about those changes, and do you think other changes might need to be made as well to sort of encourage cell therapies?
I don't know as much about that as I should, so I'll make a general comment, which is the intention of that act is, I think, a very good one, is to say, as we move into making new kinds of medicines, which are not chemical toxins that kill cells, but rather using, let's call it nature's solution, natural cells to use to treat patients, one should rethink the regulatory constraints without, and I want to emphasize this, without in any way sacrificing safety as a major concern. So I think it's a bit too early to know what will be the long-term consequence of that, but I think it's fair to say that people here at the meeting think that that's a generally good idea, is to say that there may be a new way to make medicines, and I don't want to call them the old-fashioned ways, but that when one thought about, as I say, putting toxic chemicals to kill cells, and you naturally need different kinds of regulations. Others may know more about the act than I do. It's an American act, for one thing, and we have two visitors from other countries, so I'm going to have to go back to George.
I don't have that much more to say about it, other than I think it is very appropriate to recognize this moment in time where there is a meaning to 21st century cures. It's not just a slogan. But I think that the Food and Drug Administration in the United States, and the equivalents EMA and CFDA and so forth, are prepared for these in that it is safety and efficacy testing as always. The small difference is in the case of editing, it's hard to do preclinical trials in animals. There's one nuance, which is that the off-target is dictated by the composition of the genome. So if you have a particular guide RNA, first of all, the guide RNA itself may not be conserved between the animal you're testing it and humans, and for sure the off-targets are not. And we've published a paper where even the difference between two patients could be significant. If you have a single nucleotide polymorphism, it can become an attractor to an off-target. In fact, we had a case where 37% of the CRISPR events were on an off-target SNP that was not present in the reference genome. So it's one small thing that makes it a little bit different, maybe harder to test 21st century cures of that ilk.
Molly?
I won't comment specifically on your question, but I think that we have seen over the past maybe half a dozen years, clinics popping up all over North America where you can go for stem cell therapy. We used to see this, in South America or Asia, but I think the challenge for the public is that it's very confusing because they're being offered stem cell therapies, and us in the field are very concerned about those because we don't know what people are actually being transplanted with. And so we're just hoping that nothing bad happens. But we have started to see some things happening. And I think this is just confusing for the public when you're offered all these stem cell therapies. And so it's just a little bit of a warning for people to try and be cautious as they go for some of these therapies and make sure that they really are being treated.
George?
Yeah, I'll just add to that you really shouldn't be taking drugs of any sort or treatments of any sort that haven't been through double-blind, placebo-controlled, randomized clinical trials. I mean, that's the ideal. I know there's many realistic exceptions to that, but there have been some exceptions that I think are unmerited, having to do with nutritional supplements and stem cells. And hopefully, while the crackdown is slowly materializing, people should use that high standard as their personal criteria.
May I add a slightly amusing comment? Is recently I was at a shopping mall and they were advertising stem cells in shampoo. And so that's sort of an extreme example. But I would, on a more serious note, like to remind people that the ISSCR, as Molly said, is very intent on providing guidelines to investigators and the public because these new medicines will come, but they have to come with attention to safety, and as George said, double-blinded experiments are really the ideal. We've had, I think, a previous press conference already on that subject, so I won't repeat that here. But I would also take this occasion to point to some of the things that Professor Takahashi mentioned in her talk, which is, you may have noted in several points she mentioned the Ministry of from the government helping move this forward. And I think that's really an important point. Japan, Britain, the US, and Canada, I think, are the countries that are really leading the way of saying, how do you make these new medicines really safe? Because that's in everybody's interest. Antonio?
I have some other questions.
Please.
So I don't have to get up 3 times. For Dr. Takahashi, the question, yesterday we heard from a group that is making universal cells. In fact, there's a company called Universal Cells. They delete, in some way, the HLA genes. So I would like to hear if you think that that is, I'm going to ask just 3 questions in a row, and then I'm going to sit down. Okay. And then for Dr. Melton, can there be a gadget, an artificial organ, which could be kind of just generally programmed to do, you mentioned cases where you don't have to put these cells in the actual organ that you're trying to treat, I guess things that secrete something. Could you make a general purpose module that people could have? And then the last question, sort of to your last point, and would be for everybody, is about the California Stem Cell Initiative, which has spent $6 billion so far, or has cost $6 billion. And whether you think it's worth it. Recently, they have sort of touted that they're getting to the clinic. Those therapies involve bone marrow or hematopoietic stem cells. So it's kind of these things came in sort of perpendicular to the purpose of that organization a little bit. So they're not iPS cells, they're not embryonic stem cells. So I just wonder if now that spending is coming to an end and might be renewed, whether you guys have a view about whether it was worth $6 billion.
Okay, let's take those in order if we can remember all 3. Dr. Takahashi.
About the universal cell, the HLA knockout cells, they knock out HLA type 1 and type HLA 1 and 2, but they put the HLA-E antigen, so they suppress the natural killer cells. So it's reasonable to suppress the immune reaction, but we should be very careful. Immune reaction is so complex, so the cell cannot completely suppress the NK cells, I think. So we should be very, very careful, and we need the data about the immune reaction of those cells, I think.
Maybe I'll take a stab at the other ones. Just that I agree with Dr. Takahashi that thinking about the immune reaction as a plus or minus thing is probably not the best way to do it. It's more of a balance. You often see a seesaw in their talks. It is possible without having perfect match to induce something called anergy, which is sufficient to reduce the immune response. And George and others have worked on thinking about what genes you need to modify in order to, let's say, abrogate or diminish the immune response. That's a kind of fuzzy answer, and it reveals what Dr. Takahashi said, is the immune system is very complicated, and it's not something we fully understand yet. Let's come to your idea of a gadget. It's not a word that I would have thought to use, but I'll tell you, it's a perfectly reasonable term, is could you put a packet of, say, pancreatic beta cells that read blood glucose and secrete insulin as a gadget in the body, and then that would replace the dysfunction or the absence of a functional pancreas? That's exactly what Sema Therapeutics and other groups are trying to do, is to make human cells, put them in a protective pouch made with bioengineers, and then have them read the sugar and squirt out the right amount of insulin. That gadget, as you put it, may last a year or a decade, and then you'd go in for a new gadget. I'm going to come to your last point, and then I'm going to invite others to comment on it, which was a good question, is was it worth $6 billion? First of all, I'm surprised by that number because I thought it was $3 billion, but maybe you're right. Oh, okay, thank you. So let's say it's $6 billion. Well, let me give you some other numbers. Next year, $30 billion of insulin will be injected into people. The cost of diabetes is $700 billion. I don't know what the cost of disease is worldwide, but I would say $6 billion is an absolutely trivial amount if you're trying to change the course of human health and make people happier and healthier. An analogy I might give is, imagine if I told you you had a hole in the roof of your house that was costing you $700 billion a year. How much money would you spend to fix that hole? Okay. I can put some other numbers on this. The NIH, I believe, will spend $35 billion next year. Again, that's a trivial amount compared to the cost of treatment and care, and I'm not even talking about the social cost of disease. So as you can see, you kind of touched a nerve with me on whether or not $6 billion is too much money. I would say, cheer on California. Let's have more states and nations do that, and that's just the right thing to be doing. Let me invite others to comment on that. I don't actually know how much money is spent in Japan on stem cell research or Canada. Molly, do you have something to say about that?
Well, I would agree with you that we should encourage more states and countries to invest in this field because we are trying to change medicine. You know, I always think that in regenerative medicine, we can go beyond treating the symptoms of a disease and stop it and reverse it. That's obviously what you're trying to do with blindness. That's an area that we're interested in as well. So it's a whole different way of thinking about treating disease. And yeah, I don't think any of us would disagree that we need, you know, more funding to change just the way medical practice is being done. It's that leap that does require investment. So, you know, we in Canada invest quite a bit in this field, but it's really small compared to what California has done. Did you want to add?
Yes, in Japan, we always complain about the research money about stem cell because it's less than 1/10 of the California research money. But recently, in Japan, big pharmaceutical companies are now interested in the stem cell treatment. So the commercial money comes into this field recently.
So if I might, Antonio, give a slightly different answer to turn your question, which would be, I think you report largely in the U.S., and I would say you, like me then, have lived through not quite a decade of discussion of who's going to pay for the health care we have. The discussion, the endless discussion about Obamacare repeal and replace has not been to find new medicines or make the healthcare better. It's been an argument about who's going to pay for it. I think as a nation, it would be much better if we talked about how to use the resources we have to reduce healthcare costs, not by arguing about who pays for it, but by finding new medicines. I'm not asking you to defend.
I have to make a comment.
Yeah.
The money was earmarked for particular technological concepts.
Oh, in California.
So it wasn't, the money's not available to anybody who might have a small molecule or something.
Right.
Generally, it's earmarked for a particular concept. And right here, we can argue here that some of those concepts have been thrown out the window, like the use of personal iPS cells from you. That turns out not to be, that's too complex. So part of the question is should money be earmarked?
I see.
Specific,
Well, almost
for a
all,
specific thing as opposed to, next door there's the meeting of the whatever society,
Right.
maybe they should get the $3 billion.
yeah, no, it's a good point, and it's a broader discussion that's probably inappropriate for today, but I would argue that almost all taxpayers' dollars is earmarked for something, and so the question is who decides on how it's earmarked. And your point that iPS cells have changed their focus, that's not because of the science, it's because of the cost. I think if you might remember Dr. Takahashi's slide, it was not that doing individual iPS cells wouldn't work, it's that it cost too much. And I'm not qualified to comment on how much costs will come down, but I think we would all agree that the cost of making iPS cells is likely to come down very significantly in the years ahead. So I wouldn't rule out the possibility that in the next generation, you're here to report on personalized medicine with iPS cells. Now, you might say, Doug, when is the next generation? But I particularly took that term because it's such a tricky term. It could mean 10 or 30 years or something. But these are very good points you raised. Thank you.